Okay, we're finishing up chapter 17 here. This is going to be section 17.8 on viruses. Now, what's a virus? Well, first let's do their little introductory thing on, this is HIV. This book was written a couple of years ago before the COVID thing was a thing. And so, but what's true of the HIV virus is true of a lot of other viruses. It's a retrovirus, and it's an RNA virus. It gets into the cell, makes its own, it gets into the nucleus, and makes DNA out of it. Uses its RNA. It's an RNA virus. Uses the RNA to by means of a reverse transcriptase, which it brings into the cell with it when it infects a cell, and it destroys T4 lymphocytes. It's one of the many kinds of white blood cells when they're involved in an immune response, and it just does things to just it inactivates or deactivates or damages the immune system pretty bad. A virus. Small particles, 3 to 200 genes, and I'm going to give you a word here. I'm going to give you a term that you probably haven't seen, but let me get my whiteboard out. Viruses really aren't considered organisms. They're really not considered living. Anyway, I've seen this term. You don't have to remember it, but it's called a supramolecular complex. Now, supramolecular chemistry it's a research area. I'm really not. Never been involved in it. I'm not really that familiar with it, but it does exist. This is supramolecular compounds. They are compounds that are, you can call them pure substances, but they are made of like several, more than one molecule that somehow functions together. And one of these, I'm going to show you one of them. You will have organic compounds. Let's say this is just some kind of a ring. It wasn't much of a ring of it uses cyclohexane. Okay, you know cyclohexane, cyclopentane, things like that. Well, make this a big ring. All right, let's say it's a big ring, and then have another ring which is kind of like a chain. All right, you make these chains. This is called a catenane. All right, well, what does this have to do with viruses? Well, a virus is essentially a supramolecular complex. You're going to have a nucleic acid. Let me do some things here. So you have nucleic acid. And you're going to have maybe other, things like maybe some enzymes that it's going to need for reproduction. And then you're going to have this whole thing in a protein coat. And the number of compounds, or the number of molecules in these things can be fairly small. Some are larger than others, some are more complicated than others, but some are really simple. Okay, anyway, that's what a virus is. 3 to 200 genes that require a host cell to replicate. So these things really aren't alive. They can't reproduce on their own. So they contain a nucleic acid, either DNA or RNA, inside a protein coat. They do not have the necessary material such as nucleotides and enzymes to make proteins and grow. They may have a few, like I said, they may have a couple enzymes of their own, but they can't do anything with them until they get into a host. And they replicate by invading a host, taking over the machinery that just take over the cellular machinery and make little viruses, lots of them. Every organism has viruses that can infect it. Bacteria have bacteriophages, and they're, in fact, bacteriophages were some of the early research on biochemistry and nucleic acids and that were done with those things, because they're, bacteria are cheap, easy to reproduce, and bacteriophages are, well, bacteria get infected with a bacteriophage. A bacterium gets infected with a bacteriophage. It's going to generate a lot of virions. Virion, V-I-R-I-O-N. That's like a virus particle. Okay. Not all viruses cause disease, but some do. We see some of the common ones here. And again, we don't have COVID, the coronavirus, I forget the designation, but it's a SARS CoV-2 or something like that. But you can find it easily enough. There's a common cold. There's over 100 types. These are coronaviruses. Coronaviruses are named because of their structure. They're a little ball with these little spikes on them to give the appearance of a crown. Or to me, it reminds me of these World War II sea mines that were, we used to mine harbors where, prevent shipping from going in and out. And then there's also rhinoviruses. There's over 110 types of those. And you've got influenza viruses. You got warts, herpes, human papilloma virus, leukemia, cancers, AIDS, retroviruses, then several others. And I think this is time that there was another thing I wanted to mention. Earlier on in this series on chapter 17, I mentioned something about the central dogma of molecular biology, which, let me get the whiteboard up here and I'll just describe it. This is why you don't talk about dogmas, okay? There's this, get this thing up here. Okay, we can, I'm going to zap this whole thing. No, it's easier this way. Control X, there we go. There we go. Okay. Remember we said the central dogma. And I think I had mentioned that it was Watson or Crick. I'm pretty sure it was Watson actually regretted using that term because it has connotations that you really don't want in the sciences, but it was DNA, RNA, protein. Okay, well, that is not correct because you have retroviruses. Which go RNA to DNA, back to RNA, and then to protein. Okay, so this central dogma has to do with information flow. But that's mostly a historical thing that I'm giving you here. Okay, and we're going to see how some of this stuff works. Okay, but the way a virus works is it attaches to a host cell, and you see the diagram there, and you could this is what they have there. Looks like a coronavirus, so it probably is. If a cold virus or something like that, that's what they look like. And what will happen is it'll land on the cell membrane. It will penetrate into the cell, and then it will break apart the proteins there. They might there might be some enzymes in there that it needs, but the main thing is that you see the nucleic acid from this virus getting into the cell, and it essentially hijacks the cell's machinery and makes viruses, makes a lot of viruses, and the cell bursts and new viruses are released to infect other cells. So what's going to happen is, okay, if a DNA virus is going to inject DNA, and then it's going to synthesize the proteins, which it's mostly the virus is nucleic acid and proteins for the most part. So it's going to synthesize, it's going to reproduce the nucleic acid, but the DNA, and it's also going to make a lot of proteins. And then what's going to happen is the virus is simply going to the virus particles are just going to come together, and then eventually the cell is going to be overwhelmed. It's going to break apart, and the new virus particles are going to be released. So you could, from the picture's worth 1,000 words, you can pretty much see what's going on here. See the virus comes in and makes make more viruses, and then the virus eventually is released from the cell, or can destroy the cell altogether. Bacteriophages will do that. They'll just the bacterial cell will just break open and just spew lots of these things. What happens? A virus infection begins with the virus landing on the cell wall or the cell membrane, and it makes an opening. And what usually happens is the viral nucleic acids enter and mix with the materials in the host cell, and eventually the host cell starts replicating the viral DNA if it contains DNA, and if it'll also produce viral DNA and proteases, to produce the protein coat. So it's got to synthesize proteins there too, and then it releases more virus particles to infect more cells. Reverse transcription is different. Retrovirus contains viral RNA, no viral DNA. The viral RNA has its own, the virus has its own reverse transcriptase to produce a viral DNA strand. And then the viral DNA strand forms a complementary strand, and then the new DNA uses the host enzymes to make more new virus particles. Okay, so you're going to end up, so it's going to inject RNA, and then it's reverse transcriptase. It's going to get into the nucleus. The RNA is going to get into the nucleus. Its reverse transcriptase is going to make DNA out of the RNA, and then that DNA is going to start generating RNA and also viral proteins via the processes that we've already seen. So here's and anyway, what's going on with this is they're using well, this is just a retrovirus here. They're not giving a specific one, but this could be something like COVID. This could be something like the HIV virus. It could be any of a number of things. So retrovirus gets into the cell and it forms the DNA, as we just saw we read on the previous slide. So you see, if we're up here, you get the viruses getting in now. Actually, the protein coat is usually left on the surface here, and it will inject its nucleic acid and other things that might be there. So you have this is a retrovirus. So you have a reverse transcriptase, produces viral DNA, and there's a couple different mechanisms where this could go. You see what's going on here. You have the viral DNA. It forms a provirus, And what it actually does is it actually gets into the nucleus, and it it actually incorporates itself into the host DNA. So remember I said, I think it was in the last, it might have been the last, I think it was the last section where you have a viral enzyme is a viral protein was actually incorporated into mammals about 50 million years ago, and it is in the placenta. It's used in the placenta now. So there's a thing that was good for the host, and well, these aren't, viruses aren't necessarily bad, but they can cause, there are some that cause disease. Okay, so let's go back and see what we had here. You have, retrovirus reverse transcriptase to produce the viral DNA, forms a complementary DNA strand, and then uses the host machinery to synthesize new virus particles. And you see what's going on here. You have virus, the DNA is incorporated into the genome. So that means once you have a cold, or if you get one of these retroviruses in you, that you're carrying the gene there in a lot of cases. And of course, some of these things have their own proteases and protease inhibitors. But what happens is when the cell replicates, when the virus replicates, it's going to make more viruses, and they're going to leave and infect other cells. We saw the HIV virus infects the T4 lymphocytes, and it essentially kills them, decreases the level, and essentially it just cripples the immune system. Your immune system is going to protect you against a lot of things just automatically that you don't even realize are there necessarily. And they mention here the pneumonia and skin cancer associated with AIDS, and they give somewhere else, there's somewhere here they talk about there's opportunistic infections is what they will allow. They don't really cause the infections, they just stop the immune system from preventing them. So there's certain pneumonias and something called Kaposi sarcoma, which is a skin cancer, one of the common things from HIV. So, and this is, you notice this is some of these things just have a protein code in the nucleic acid. This thing has this is a little more complicated, lipid membrane. It's got there's reverse transcriptase in there, and there's the RNA, and then a core. So there's actually a few things in it, but they're still reasonably simple compared to things we consider living. How do you treat these things? Well, what you have to do is attack things like HIV at different points in its life cycle. So you prevent the reverse transcription of the viral DNA. And then this is one of the AZT, similar to thymidine, and it mimics the structure of the nucleosides used for DNA synthesis. We're going to see the structures of these. So what happens, you remember the lock and key enzyme picture? Well, if you have something that's going to recognize thymidine and bind to it, or like if it's an RNA molecule, DNA molecule, or DNA strand, RNA strand, this thing will bind to it and it can't react any further. So it essentially inactivates that particular enzyme, or that stops that particular process. Then there's protease inhibitors that will prevent protein synthesis used by viruses. They have some of these things have proteases in them, and you inhibit those, and they can't make these anymore. And in fact, there is a I can't think of the name right now, but there is an experimental antiviral for COVID, which is only experimental, and it does contain protease inhibitors, which of course they're going to prevent these proteases from doing what they need to do to help the virus make more copies of itself. Anyway, here's a couple of these things that Lexiva is a protease inhibitor. The azidothiamidine AZT. You see there's this azido group here. This looks like it looks a lot like deoxythymidine, okay? Except it's not. It's azido. It's not. It's this. This isn't a hydroxyl here. So if this thing gets into at the point where this is going to be incorporated into a DNA molecule, this azide is going to stop that from happening. And this Lexiva, it will prevent the protease from doing its work. Okay, I actually have, there's a something called Paxlovid, which is a combination of nirmatrelvir, which is a what is it here? It's what does it do here? I can't find what it does. Just the chemical name, structural formula. And then they have one called ritonavir, which is it is a protease inhibitor. So there's certain proteases, they're going to cleave proteins. They might, for whatever reasons, they might make a big molecule, and then you have to cut pieces out of it in order to get this thing to maybe assemble in a certain way, or form the protein coat or some such thing. Anyway, it's there's a couple of okay. There's the nirmatrelvir is a protease inhibitor, and it says it renders the COVID virus incapable of processing polyprotein precursors, preventing viral replication. And the ritonavir is a protease inhibitor, but it's not active against the SARS. It inhibits it inhibits another metabolism. It inhibits the metabolism of the nirmatrelvir, and see if I can get a copy of this at some point. But anyways, you have one protease inhibitor that's inhibiting the protease that forms virus particles, and then you have another protease inhibitor that inhibits the action of the enzyme that breaks down that protease inhibitor. So you've got two protease inhibitors in there. It gets complicated, but these are a couple of them, and you see these are two antiviral drugs. Okay, learning check. Match the following terms with the description: virus, retrovirus, protease inhibitor, reverse transcription. Okay, well, a virus containing RNA is a retrovirus. Okay, a small particle requiring a host cell to replicate, that's a virus. Okay, needs a host cell to replicate. Substance that prevents the synthesis of viral proteins, that's going to be a protease inhibitor, I guess. Using viral RNA to synthesize viral DNA, well, that's going to be reverse transcription. So match the following with the following. Okay, retrovirus, virus, protease inhibitor, reverse transcription. Okay, that's the picture there. And we're getting near the end here. Link to cancer, where you've got an Epstein-Barr virus, which is can cause herpes virus 4, which can cause cancer in humans. And what cancer basically is a uncontrolled, ceaseless cell division. When there's see, cells have a machinery in them, have a mechanism in them where if they run into a cell of another type, they're going to stop dividing. Okay, cancer cells don't do that; they just keep dividing. And they're also in There's also what they call telomeres, that when the DNA, every time the cell divides, the length of the DNA is reduced some, and once the telomeres have been removed and you're actually getting into an actual gene, there are mechanisms for programmed cell death. And when those things are short-circuited, or you have there are enzymes that will add telomeres, they will just keep reproducing forever, and that is not a good thing, as we saw. Okay, there's chemical and environmental substances. They can mess with the DNA, and there's a lot of these things: aflatoxins, aniline dyes, arsenic, asbestos, cadmium. And I'm sure you've seen all these things, and you already know that all these things are bad. And usually, okay, these like cadmium, chromium, nickel, those are metals, and the 5-cent piece has a lot of nickel in it. But these usually, I'm not going to say necessarily for certain, but when you're in a certain form, like, well, cadmium is used in a lot of oil pigments for oil paints, but it's insoluble. The pigment's insoluble. It doesn't really react. It's not going to, unless you don't want to ingest it, okay? But it's really not going to go anywhere. If it gets on your skin, you wash it off and you're okay. If you get a soluble compound of these things, then it gets into your system. Then it can go anywhere it wants. Then it causes a lot of problems. Okay. So anyway, those are some examples. All right, we're done with section 17.8, and I'm going to give this in lecture. If you don't see this beforehand, or you're going to have it to refer to with or without the lecture. So I'm going to get this up onto YouTube, and you can have at it.